The structure and the function of the nucleus. So the nucleus is the largest organelle inside any eukaryotic cell, and almost all eukaryotic cells have a nucleus. One of the only eukaryotic cells that doesn't have a nucleus is red blood cells. Let's talk first about its function. So the function of the nucleus is to retain the genetic material of the cell. And we know that the genetic material in eukaryotic cells is DNA. So essentially, the nucleus contains the DNA. The DNA, we know, codes for proteins. And the section of DNA which codes for a particular protein is known as a gene. Genes are split up into two sections. They're split up into introns and exons. The intron is the non-coding region, the section that doesn't actually code for amino acids that make up the protein, and the exon is the expressed region, the region that does code for the protein. Together, these are known as the cistron. So the word cistron and the word gene are somewhat interchangeable, although the more common usage is gene. So this DNA, you probably are most familiar with DNA in the form of chromosomes, so this structure that we often draw, this sort of X-shaped structure. So this is a chromosome. However, for most of the life of the cell, when it's not undergoing mitosis or meiosis, it doesn't actually look like it's in this X-shaped structure. It's unfolded and unwound. So it's in a form that we refer to as chromatin. I know I'm introducing you to a ton of new words here that you might not have come across before. So the main, most basic function of the nucleus is to retain the genetic material of the cell in the form of DNA, which is used during the most of the life of the cell in the form of chromatin. The nucleus does have a couple of other functions, and let's just talk about those for a second. So one is the production of mRNA and tRNA during protein synthesis. The production of proteins. This production of mRNA is known as transcription. Number two, the production of ribosomes. So ribosomes are made up of protein and rRNA. This is done in the nucleolus, a rather dense section of the nucleus that you'll see under the electron micrograph. So let's talk about structure of the nucleus. So right in the very center, we've got the nucleolus. And then we have got this double membrane. Rather dreadfully drawn there. Now this double membrane has got nuclear pores in it, so it's got small holes. These nuclear pores are to allow the movement of proteins, mRNA, enzymes, and other really important things to move into and out of the nucleus, but to not allow the movement of DNA out of the nucleus. So DNA is trapped within the nucleus, but some things need to be able to move in or out of the structure. So we have these nuclear pores. These nuclear pores are actually really complex little pieces of machinery. And if we were to look at the diameter of a nuclear pore, we would see that they're about 100 nanometers in diameter. But strangely, because they're quite complex little structures, they've got a lot going on in them, they'll actually only allow things to travel through that are about 9 nanometers in diameter. So they only allow really, really tiny things to move through. The other thing we might expect to see in a nucleus, if we were looking under the electron microscope, is the chromatin, so the unfolded and unraveled DNA. Just one more thing about the nucleus before we finish, and that is the outer nuclear membrane is continuous with the rough endoplasmic reticulum. So the rough endoplasmic reticulum grows out of the nucleus. It is continuous with the nuclear envelope. So I'm going to draw a pretty rubbish diagram here, but if we were to imagine it growing outwards. And that's it. That's basically everything you need to know about the nucleus at this point. Well done.